Yeah, please do let me know if anybody has any issues with the audio or something. So anyway, let's see. So these are the topics that are left: cellular senescence, stem cells, and interventions in aging. So all these three have been, very nicely brought together in today's lecture. So we will be seeing that most of interventions also they involve targeting this senescence, which is very closely, sometimes interchangeably also used with aging. So with age, what happens? The senescence cell, they increase in number. So anybody could tell me what is senescence? No one?
Cell death, like routine cell death kind of.
That is apoptosis,
Okay,
programmed cell.
wait. Sorry. Accent also.
There's something called quiescence. There's something called senescence. What are these?
Oh, like after age, like it was something related to age, no?
It is related to age, definitely. That's why you're studying it. So they do not have permanent proliferation arrest. Yeah, am I audible?
सर माझे किच्छु शोना जाएंगे.
Okay. So senescence means permanent proliferation arrest. So there is no more proliferation. It could happen due to a lot of factors like DNA damage, repair, might cause cell cycle arrest by activation of P53, as you can see here, or telomere shortening, as we already know, it's again an hallmark of aging that could also lead to DNA damage repair processes that causes cell cycle arrest. Then there could be, oncogene activation that could increase the amount of reactive oxygen species that is present in a cell, and again that DNA damage repair process will again lead to cell cycle arrest. So there are several features of senescence. Specifically, there are these some features are called as SASP. SASP is senescence associated secretory phenotype. So these are particular phenotypes that are exhibited by these senescent cells. They sometimes, secrete certain molecules that cause, even their neighboring cells to become senescent. So something like that happens. Apart from that, there are some morphological changes that are involved. Some, like P16, P21, these proteins are upregulated, leading to cell cycle arrest. There is increase in oxidative damage due to increase in reactive oxygen species. Since there is an upregulated BCL2 family of proteins, which is an anti-apoptotic protein. So they are also resistant to apoptosis. So also metabolic changes are observed in these kind of cells. So in this figure, you can see how do these, senescence associated secretory phenotype, they are regulated. So there are multiple processes. One of them is the mTOR pathway. As you can see, it's an autophagic process, an mTOR, which leads to an mTOR pathway. And there are some interleukins, which are chemokines. These are also involved in regulating senescence. And the one of the major pathways involved here is the NF kappa B. Again, something very close related to cancer also. So these are essentially regulated through various routes. So I'm not going into much detail about it, but this is all about it. Then coming to the effects, there are some beneficial, some deleterious, and some ambiguous effects of senescence. So some beneficial effects are like They are important for embryonic development. For example, these MMP2 and MMPs are essentially matrix metalloproteinase. These are important for embryonic development. Platelet-derived growth factors, we've already discussed about it. They are important for wound healing, and some tumor suppressor proteins are also, seen in senescent cells. So that is one side of senescence. On the other hand, it also causes tumor development, chronic inflammation, because interleukins, chemokines, they induce inflammation. And then stem cell exhaustion is also one of the reasons, one of the things that happen. And some, ambiguous things, like sometimes, some cells, they reprogram themselves. They behave differently because of this senescent phenotype. So that is not very well known. Why does that happen? Whether it is good for the cell or not, but that is also an effect. So these are the few things that we see. So to combat this problem, there are two ways. Am I clear from till this point? Anybody could maybe answer? Antik, you're not audible today. I don't know why. Still not audible. Anybody else can hear Antik? Maybe I'm not able to hear him. I don't know what's the reason. There is no response. At least I don't know what's the issue.
Sir, Raji ki phone in the ashram.
I think he will be joining us today. I'm not sure. Let's see. Previous slide, is this the one, Antik, you want? I have to start with. Yeah, so this explains the various effects of senescence, the consequences of cell senescence. So while there are senescent cells that are important for certain functions, like these are certain proteins which are produced by senescent cell, which are important for embryonic development, which are important for wound healing and tumor suppression. On the other hand, there are again certain things which are expressed by the senescent cell that promote in tumor formation and chronic inflammation and stem cell exhaustions, things like that. So there are both roles of senescence. Not very well understood what exactly happens in each context. So there is a context dependent function of senescence. Is that clear? Antik has to type today. Okay. Anything. So to combat this problem, there are two approaches. One is senolytic approach, where we kill the senescent cells. That is one of the approaches. So what happens is we use various pathways, like the mTOR pathway, which was discussed. So earlier, mTOR pathway is very important in terms of aging. Anybody knows what is the full form of mTOR? Anybody?
Mammalian target of rampicin or whatever. Google
Rapamycin,
the
right?
also,
Right.
sir.
Okay, that's good. So that is exactly what it is. We'll talk about it. So, like mTOR pathway is affected using certain flavonoids, like quercetin, fisetin, dastanib. These are some flavonoids which affect this pathway. Particularly, so if this pathway is affected, these cells could be killed selectively. There are certain pro-drugs also, senolytic drugs, which cause killing of these senescent cells. And then there are certain drugs which inhibit the BCL 2 family proteins, which are anti-apoptotic proteins. They will cause, again, selective death of these kind of this cell. So similarly, HSP19 is deacetylase. So preventing deacetylation also, it has been observed that they also cause selective killing of senescent cells. So that is one approach. Another approach is to, modulate the senescent cells to only exhibit the beneficial functions, not the deleterious functions. So there also there are drugs like rapamycin and all these other things which are involved, which works in a similar fashion like senolytics, but they do not cause the cell to die. Rather, it prevents the cell to exhibit some harmful senescent phenotypes. Is that clear? Please give some reaction. Maybe that will be easy for me to understand. Today we have a lot of things to cover up, so I'll be a little bit fast. Antik, yes. Okay, I think it's clear to you. Okay, mTOR, regarding mTOR, we were talking about this pathway for this is involved in various, pathways. Like stem cell turnover, cellular senescence, inflammation, protein translation, autophagy, all of which are, linked to aging. So this pathway is a very important pathway for us. If we can control elements of this pathway, definitely we could control aging. Yes, Srinjan.
So stem cell turnover টা কি মানে কি হচ্ছে
How many stem cells are there? They are produced. So stem cells are also produced from stem cells. So they are also, this pool is replenished. So that is exactly the turnover thing. Clear?
মানেটা কি turnover এর?
কত প্রডিউস হচ্ছে?
আচ্ছা, means they are like mTOR is producing the stem cells.
No. It is involved in this is a pathway that decides that how much stem cells will be there. We will talk in detail about stem cells. So how much stem cells are present in the total stem cell pool, that is decided by this mTOR pathway.
So one more question, sir.
Hmm?
So cancer cells are stem cells, right? Right, as of now, we can consider it. So
They are similar to stem cells. We will see how that happens. Yes.
so this mTOR is also responsible for increasing the number of stems, like cancer cells as such.
Actually, they do. We will talk about it. Actually, we are coming to stem cells very soon. We will answer those questions there. That's a good question, though. For obviously mTOR, it stands for mammalian target of rapamycin. Hence, rapamycin is the drug that is used. And in mice models, it has been observed that rapamycin has increased the longevity of mice models. As you can see here, rapamycin and resveratrol, these are rapamycin analogs. They increase, the longevity of organisms, at least model organisms, it has been observed. But there could be some side effects because mTOR pathway is involved in a lot of processes, lot of key processes. So we just cannot just take rapamycin or resveratrol just like that and expect that we will live a long life. Although resveratrol is something that is also present in red wine, so which also has antioxidants, which is important for longer life. But again, there are various contradictory studies on this. I'm not going into it. But the thing is that rapamycin can increase the lifespan at least slightly. It has been observed in aged mice when it was done. So even gene therapy approaches were also tried in aging. Yes, Chinjan.
So sorry to interrupt, but can you once again go to the previous slide? So why is there a difference between male and female in the survival chance?
That I'm not sure. There could be I'm really not sure why is that so. I have to read about it. Sometimes they just work like that. So maybe the hormonal things might be involved. Maybe something to do with testosterone or something like that. I'm not sure.
So can you give one more example like this, where we can see like this thing?
Male and female bias. Usually, when I was doing studies with mice models, I used to only use male mice because so there are sometimes these there are some changes in hormone which lead to a difference in these kind of studies. Even if you see the proportion of lung cancer happening in men is far higher than that of women, even in case of certain things like that. In humans also we see such kind of differences.
Can we
So
call it like sex influenced thing?
yeah, of course, sex bias is definitely there in a lot of things. So like
Means
breast
it's
cancer
related to
obviously
genetics,
happens.
something. That's what.
Yeah, yeah, yeah. Genetics is one of the reason. Like
Mhm.
breast cancer only happens for females, prostate cancer only happens for males. But these organs are present in both the sexes, like that. So again, sometimes sex does influence. But I think this particular study has something like this. They have done studies on Drosophila also. They have seen, so on various model organisms they have seen an increase in lifespan due to rapamycin, although it's very minimal, but it's there. So some gene therapy approaches could involve in vivo gene therapy. That's how you will just put the gene right inside the cell, like through liposomes or viral vectors. So your viral vectors could be adenovectors or retroviral vectors or lentiviral vectors. We'll talk about it in detail. There could be polymers. There could be nanoparticles as well. There could be some peptides called cell-penetrating peptides, which can be used for gene therapy. And for ex vivo gene therapy, that is essentially you take the cell from the body and then you infuse the genes into them. So you transfect them and the genes are present. Now you grow these cells and then you put these cells back into the body. That is exactly ex vivo gene therapy. Some promising approaches are there, but again, this is not present in the clinical stage as of yet. So regarding the um just wait a minute. Regarding the viral approaches, like adeno-associated viruses, they will contain the DNA which is required for the protein you want to be produced, and then it goes inside the cell, and then it goes inside the nucleus. This is how the nuclear pore looks like. So the virus disintegrates outside the nuclear pore. Your DNA goes inside the nucleus, and it causes whatever it needs to do. So that is one thing that could happen. So in mice, it has been observed that telomere expression have been, genetically increased to increase the lifespan of mice. And again, it was observed that it was successful in mice. But again, this kind of gene therapy to do in humans is we are not yet ready for such things yet. So in terms of clinical trials, this is not in a very good state. So
So why telomere?
telomere attrition is one hallmark of aging. So if you have telomerase, what will happen? It will increase the telomere length. No,
Okay, okay, got it.
that's very simple actually. So experimentally, it has been done, and they have seen that mice they have an increased lifespan. So that is one thing. Then we are coming to slowly into this topic of stem cells. So this is exactly the way how an organism developed from fertilization, formation of a zygote, to formation of a blastocyst, and then there is the embryo, fetus, baby, and you grow old and things like that. So before in the zygote stage, when you have like one cell or two cell, four cells, something like that, at that point, you have totipotency. Those cells are totipotent cells. Then in the blastocyst, there is something called the inner cell mass. These cells are pluripotent cells. And then when you start growing, then you have multipotent cells, unipotent cells, which are called progenitor cells as well. So with as you start aging, as you start maturing, the plasticity of the cells increases, the potency decreases. So anybody can tell me what is the difference between totipotent and pluripotent cells? Just quickly. Antik is trying to say something, but we can't hear him. Anybody else can hear him? Oh, oh. Nobody? Totipotent and pluripotent, the difference?
So इस ग्राफটা দেখে লাগছে কি লাইক পোটেন্সি কমে যাচ্ছে, দ্যাটস ও ছাড়া তো বুঝতে
That's
পারছি
true.
না।
That's true. Oh, oh. Antik has written something. Let's see. Totipotent can divide into any type of tissues, and pluripotent can divide. He's typing and I'm literally reading it. Let's see what he has to write. Let's see. There is one particular organ that is very important. So anyway, for stem cells, they could be embryo derived or adult stem cells. Essentially, these two stem cells are mostly the stem cells that have been studied. So embryo-derived stem cells are derived from embryo, like embryonic stem cells. It could come from a stage before that also, a stage after that also. But mostly the cells that are used, embryo-derived stem cells, those are embryonic stem cells. Similarly, adult stem cells, these are actually uh, multipotent cells. They are not pluripotent cells like embryo-derived stem cells are. So they are like hematopoietic stem cells or mesenchymal stem cell, neuronal stem cells, things like that. So for each tissue type, there is a stem cell which is present in your body. So we take stem cells from there and we can then regenerate new cells. So that is the idea. The problem with embryo-derived stem cells is that it is an invasive process. It causes there are a lot of ethical issues that are involved when you are dealing with embryo derived stem cells, but adult stem cells don't have such issues. But again, the potency is definitely reduced in adult stem cells. So to combat this problem, there is a way that is called the induced pluripotent stem cells. So that was discovered into around 2006. So we'll talk about it. So what happens in case of stem cells, embryonic stem cells, they come from the inner cell mass of the blastocyst. We know this. Uh, Adult stem cells, they come from the adult cells. Within the adult cell, there is a pool of stem cells. There are few in number, but they are present even when you are adults. So these cells have the regenerative capacity. So whenever your cells are dying, so at that time, these cells will divide, differentiate, and replace those dying cells. That is the function. And then there is a very promising role. Any somatic cell, and you could reprogram them to become stem cells. That is what is induced pluripotent stem cells. So have you heard about it? Anybody? Just quickly, yes, no. Shinran, am I audible? This is very difficult in an
Yes
online
sir, yes sir,
class.
yes sir, yes sir, মাইক অন হচ্ছে না, বলতে পারছি না।
Okay, so then again, for stem cells, if we see the embryonic stem cells, they are pluripotent, they have infinite self-renewal capacity. The disadvantage is ethical issues, and they are immunogenic. Immunogenic in the sense that, if you cannot actually take, Mayuk, any questions?
No, sir.
Okay. Immunogenic because you cannot take, embryonic stem cells from, I mean, you have to store it. So now there are some stem cell banks which store stem cells right from the embryo stage, but otherwise you have to take it from someone else, so there would be immune problems. For adult stem cells, they are, good. There is no tumorigenicity in these stem cells, and they are usually autologous. So there is no issues of immune rejections. But they are not pluripotent; they are multipotent, and there is limited self-renewal capacity. And again, it is difficult to isolate these cells. iPSCs, on the other hand, they are pluripotent and they can be produced from adult cells, normal cells, not stem cells, adult stem cells, normal cells, and they are not immunogenic also, and they have no ethical concerns as well. But one problem that we still deal with induced pluripotent stem cell is their low reprogramming efficiency. So very low number of cells out of maybe 100 cells which you are trying to reprogram, maybe 5 of them will actually become iPSC. So that's a very less efficient method, at least till now what we have. So this is what totipotent and pluripotent difference is. So the fertilized egg and then few other stages actually are totipotent. Totipotent cells can form the placenta, while pluripotent cells, they cannot form the placenta, but they can form the other 3 germ layers of the cell. So that is the idea. Multipotent cells, they can only form tissue specific, they can differentiate in their specific tissue niche. And then there are unipotent cells which cannot differentiate further. So that is the idea. So these are the examples. What are the totipotent cells? What are the pluripotent cells, multipotent cells? And then again, progenitor cells is one another term that is used for multipotent cells. Uh, and then there are unipotent cells. You can go through this slide later. So as we said, as we differentiate further in the cycle of a cell, the plasticity it decreases. So that's what is observed. And for a progenitor cell, it's a biological cell. It's like a stem cell that has a tendency to differentiate into a specific type of cells. Uh, like we have hematopoietic stem cells, we have neuronal stem cells, we have mesenchymal stem cells. These kind of stem cells are also called like hematopoietic progenitor cells, particular cell lineage. So they are not exactly pluripotent. So this is the difference. Then this is how the placenta looks like. So as we already said, totipotent cells, they can form the placenta, while pluripotent cells, they cannot form the placenta. So for those who do not know what a placenta is, it's an organ that connects the developing fetus with the mother. To the uterus of the mother, and it allows nutrient uptake, thermoregulation, waste elimination, gaseous exchange. It also is involved for some antibodies which are transferred from the mother's womb to the infant, and some hormones, they are also transferred through this placenta. Totipotent cells can form placenta. Pluripotent cells cannot form placenta. I'm just repeating this again and again so that you guys have this in mind. So these are some definitions. What is totipotent cells? What is pluripotent cells? Multipotent cells, progenitor cells, and unipotent cells. I've already talked about it. Not going to again go through this. So you can read it by yourself. Then embryonic stem cells, they are undifferentiated cells. So this is exactly at the stage where the cells, they have not differentiated into any particular cell type, and they have unlimited self-renewal capacity, and they are pluripotent. They can form any of the 3 germ layers. So, so the 3 germ layers, again, they are the ectoderm, mesoderm, and endoderm. We'll talk about it in details. So the embryonic stem cells or the induced pluripotent stem cells, they have this pluripotency where they can form the any of the 3 germ layers. So the ectoderm, like the skin cells, nerve cells, retinal cells, these all form the ectoderm. Heart cell, blood cell, muscle cells, they form the mesoderm. Pancreatic cell, lung cells, liver cells. So these are 3 different germ layers. which a pluripotent cell can form. So do we actually need a pluripotent cell or a totipotent cell? Anybody would like to answer this? For therapeutics, I mean. No answers yet. Would we like to have a pluripotent cell for therapeutics or a totipotent cell?
Pretty potent.
Totipotent. Why?
Because our generation বেশি হবে.
Actually, pluripotent or totipotent just difference টা হচ্ছে যে totipotent can form the placenta, pluripotent cannot form the placenta. So people have tried to produce totipotent cells also. Very difficult to get them actually, because these cells, they actually do not have any chance of developing a placenta. We do not need a placenta in adult life. So we produce pluripotent cells. That is the reason why we do that. And
তো
there
বোন
is a
ম্যারোতে কোনটা থাকে? বোন ম্যারোতে কোনটা থাকে?
bone marrow, we have adult stem cells, hematopoietic stem cells.
Okay. Got it.
So these are all the differences, ectoderm, mesoderm, endoderm. There are something called germline cells also, which are actually important for reproduction. Apart from that, you can see all these three kind of, germline. So all the tissues, all the cells, they are formed using these three germlines only. Placenta is no longer required. So pluripotent stem cells are enough to, uh, complete our requirement of whatever therapeutics we need from stem cells. So one of the hallmarks, if you remember, of aging was exhaustion of this stem cell pool. So if we can give stem cell, if we can induce pluripotent stem cells, and we can, we can capacity again. Am I audible? Hello.
Yes sir, but মাঝে কিছু শুনতে পাইনি.
Yeah, so as I said, I was telling about ectoderm, mesoderm, endoderm, how these three germ layers, they comprise all types of cells you have in your body. So if we can induce pluripotent stem cells, then we can actually, if these, any of these cell types are not present, they are less in number, you can replenish their numbers using pluripotent stem cells. That is the idea. As you might have remembered that, stem cell exhaustion is one of the hallmarks of cancer. Sorry, hallmarks of aging. So if we can, induce these stem cells and we can reinfuse them in your body, they can, in theory, infinitely keep regenerating cells. And that is the idea behind stem cell therapy. Knows how iPSCs are produced? How are induced pluripotent stem cells produced? He's trying to say something, I don't know. আমি আজকে রিয়েলাইজ করলাম আন্তিক কথা না বললে মানে কিরকম একটা লাগে, মনে হচ্ছে কেউ শুনছেই না। Anybody else? Mayuk?
Sir, could you please repeat the question once more? Was it something about
Okay, it was, the question was, what was the question? I forgot the question in this entire thing.
how are pluripotent cells produced or something?
Yeah, that's the question. How are pluripotent stem cells produced? Induced pluripotent stem cells, iPSCs, how are they produced?
Uh, Pluripotent cells are basically, I think it's some kind of alteration in cell division or growth
Yes,
that
yes,
produces
yes,
pluripotent cells.
yes,
That's
yes,
what
yes.
happens.
স্কিন দিয়ে পাই তো।
স্কিন দিয়ে আমি তৈরি করছি, কি দেবো ওটাকে যে ইনডিউস প্লুরিপোটেন্ট স্টেম সেলস হবে?
কি দেবো মানে?
স্কিন সেলস নিয়ে নেবো আর ওটাকে গ্রো করলে কি ওটা আইপিএসসি তৈরি হয়ে যাবে?
ও আচ্ছা, সেটা।
এই ডিসকভারির জন্যই নোবেল প্রাইজ পেয়েছিল 2012-তে। উই উইল টক অ্যাবাউট ইট। দিস ইজ অ্যাবাউট দি
এটা অনেকটা টিস্যু কালচারিং এর মতো কি? যে স্কিন সেল দিয়ে নিচ্ছে, এটা আবার বসিয়েছে, এটা টিস্যু কালচারিং, রাইট? ওটাই এখন আমি পলিল্যাকটিক অ্যাসিড দিয়ে করছি তো।
না, polylactic acid is I think used as a scaffold as far as I
হ্যাঁ, সে
cellular
রাতুলদা যেটা বলছিল।
cellular reprogramming is the answer. Yes, Antik, you're right. He is still answering. রাতুল বলছিল হ্যাঁ polylactic acid হচ্ছে দিয়ে scaffold তৈরি হয় যেখানে cells could grow. They cannot replace cells or
বাট
tissues.
ওটাতে তো ব্যাকটেরিয়াও এসে যাবে, ওটাকে আমি স্টেরিলাইজ কিভাবে রাখবো?
We have to use certain materials which has potent antimicrobial properties so that bacterial growth is inhibited. So but this problem is there, that bacteria, certain class of especially gram positive bacteria, they grow in nosocomial settings in these kind of scaffolds and implants. So এর জন্য অনেক রিসার্চ হয়
হ্যাঁ, সব
actually.
খুব তাড়াতাড়ি ডিগ্রেড করে যায় এটা।
Hmm. So let's talk about the limitations of embryonic stem cells. We already talked about it. Ethical issues are one of the most controversial things about this. Tumors could be formed because these are the cells which are from the very initial stage. They are not differentiated at all. So they could form any kind of cells and they could form tumors. Immune rejections could happen. So one of the answer is iPSCs, but still there is a long way to go. So iPSCs were first made by this Professor Shinya Yamanaka from Japan. So he took adult fibroblast cells, skin fibroblast cells, and gave some reprogramming factors, which are essentially transcription factors. So he used these four factors, OCT4, SOX2, KLF4, and CMYC. And then he generated induced pluripotent stem cells. Now, these induced pluripotent stem cells can form any kind of, they can differentiate into any cell lineage. This is the benefit of these kind of stem cells. So, these are undifferentiated cells, induced pluripotent stem cells. They're undifferentiated. They can self-renew indefinitely in vitro, in theory, and they can differentiate into all the three germ layers uh, that could be, that are there in our body. So any somatic cell, they could be reprogrammed into a pluripotent state. So that is the beauty of this entire process. And this is the reason why Professor Shinya Yamanaka, he won the Nobel Prize in 2012 for iPSCs, for creating iPSCs. And these transcription factors, these particular four transcription factors, which cause formation of induced pluripotent stem cells, are therefore called Yamanaka factors in his name. OCT4, SOX2, KLF4, and CMYC. Anybody knows these transcription factors from any other class or something? Is it clear? Or is it still confusing? Yeah, Antik is typing. I have to still rely. Show the
Yes, sir.
factors. Can you show the factors? OCT4, SOX2, KLF4, and CMYX. It is already there on the slide. These are the same factors in the previous slide also. So these are called Yamanaka factors on his name. These are transcription factors. One other thing about these factors are that you people couldn't answer was that these factors are also potent, tumorigen. They have tumorigenetic potential. So these factors can are also sometimes involved in tumorigenesis. So sometimes in the process of creating stem cells, we can create cancer cells as well. I think that answers your question, Shinjon. You had asked previously.
Yes, sir. But still
So
totally not clear. That's all.
it is not clear.
পুরোটা না, এখনো কোশ্চেন আছে কিছু।
So please, we will answer those questions. Let's see, let's complete this. So Yamanaka, he did his studies on adult human fibroblast by some factors. And then again, Thompson, he tried to replicate this using certain other, factors, like he used OCT4, SOX2, NANOG, and Lin28A. These are also transcription factors. These are called Thompson factors to induce pluripotency in the fibroblast cells. So if you see what are the advantages of this process, there is no destruction of the embryo. They become a source of pluripotent cells. There is unlimited self-renewal. It generates autologous cells. So we can take cells from your own tissue and we can generate iPSCs for you. That is a benefit, and it's a non-invasive source. So for taking embryonic stem cells, you have to, it is an invasive process, but in this case it's non-invasive. So what are the challenges? It's tumorigenic potential. So that is one of the disadvantage. So it may differentiate into a cancer cell. Their reprogramming efficiency is very low, very low, like 0.01 to 1%. That is a very low number. The reprogramming kinetics is also very slow. So for humans, if we want to create iPSCs, it takes 21 to 28 days. That's a long period of time. So you have to grow these cells. You have to give the reprogramming factors, and you have to monitor these cells. So it's expensive. It's slow. Which cells to use? People are trying with different kinds of cells. There is not a particular consensus on which particular cell type is better and which method is used. Again, clinically, still now, stem cell therapies. I mean, nowadays it is being experimentally done in the US, but not very popular again because they are very expensive. Um, it could cause genomic instability because when you are dealing with these cells in vitro in tissue culture setups, they might have some different form of growth than what happens in your body. So these are some issues which still come up when we are dealing with iPSCs. So can you see the A panel is the fibroblast and the B panel are the iPSCs? What is the difference? Can you see a difference in the cellular morphology? Here they are elongated cells. If you see like this, these are fibroblast cells. Here they become a little bit rounded. They no longer remain, they do no longer maintain that morphology. So that is how you understand that stem cells are produced. There are certain markers also which are used. So for delivery of these transcription factors, sometimes retroviral or lentiviral vectors are used, which get inside the cell. They then they the viral I mean DNA that gets inside the nucleus, and some they are integrated into the nucleus. So these are some viral approaches for gene delivery for reprogramming. So there could be integrating viruses like retroviruses or lentiviruses, or non-integrating viruses like adenovirus, adeno-associated virus, or Sendai virus. So this is a part which usually comes in gate most of the time. Please remember this or have this slide with you. So this is important not only for stem cells, but any kind of gene delivery. Any kind of gene delivery, the viral approaches are either integrating viruses or non-integrating viruses. So anybody can tell me what could be the disadvantage of integrating viruses?
স্যার লাইক আউটকামটা আমরা আগে দিয়ে জানতে পারবো না, বিকজ পুরো র্যান্ডমলি হবে জিনিসটা।
Integrating viruses, they integrate their self into some area of your genome, into your genomic DNA. So if it integrates itself into a very important gene, then it is harmful, right? Isn't it?
Yes, sir.
So that is one disadvantage. Non-integrating viruses, they do not integrate into your in your genome. They rather replicate separately. So like adenoviruses, adeno-associated viruses, like if you have taken Covishield, that was the adenovirus vector was used for delivering the gene for spike protein. So if you know, do you know about this? Have you read a little bit about it?
No, sir.
You don't know how Covishield was, what is the technology behind Covishield? COVID vaccine?
না স্যার পড়াইনি এনকেজে স্যার।
এগুলো তো নিজের থেকে পড়তে হয়, এনকেজে স্যার কেন পড়াবে? তো their adeno
না না বলে স্যার
chimpanzee
এসব লাইক
chimpanzee adenovirus was used, and the spike protein was, the gene for spike protein was inserted into it, and then it was, transferred to your cells. First time in such a large scale, this kind of viral approach was used for gene delivery. So you have used a gene delivery, most of you, I hope they have used the gene delivery approach. If you have used the Covishield, Covaxin used an inactivated form of virus. That's a separate thing. So what are the disadvantage of using an adenovirus? Buddy. Am I clear or is it all going above your head?
is going a bit fast.
Okay, because I have to cover a lot of things. I think it will be over very soon. Let's see. I'll be a little bit slower from now. We might not be able to separate the genome of the virus. Okay. Is it for non-integrating the problem is, development of resistance. So if you use non-integrating viruses for a long amount of time, so let's say we have already used chimpanzee adenovirus for delivering the spike protein of SARS-CoV-2. If we use the same delivery machine again and again for delivering different, vaccines, what would happen? Your body will recognize it, your immune system will recognize it as a foreign source, and it will start destroying the, adenoviruses. Hence, the gene delivery approach will be unsuccessful. Okay, that is the idea. So this is exactly how it looks like. So adenoviruses or adeno-associated viruses or Sendai viruses, they produce integration-free iPSCs, which is important because if there is some random integration into some important gene, it might lead to some deleterious effect. That is the idea. So you do not want your, transcription factors to just getting integrated into the genome, right? Anywhere in the genome, you can't, take that risk. So this is something which is written about all of this. You can read about it when you get the slides. Then there are some, advantages, some disadvantages of different kinds of viral based approaches. Again, you can read about it. Not going to go through it in detail. Then there are some non-viral approaches also, like plasmids. There are mini circles, which is essentially the same thing, a little smaller plasmid. Then there are transposons and episomal approaches. So plasmids are, these are vectors. You know about plasmid. You know about plasmid, or you don't know about that as well? ওটা তো এনকেজে স্যার পড়িয়েছেন নিশ্চয়ই। Anybody?
So can you repeat the question?
You know about plasmids?
Yes, sir.
So plasmids could be used. Then there is mini circle, which is very similar to plasmid, which could be used. So plasmids, they have, the selectable markers, they have the promoters for expression. These kind of things are present in a plasmid. You must be knowing about it. So plasmids are sometimes used. to create iPSCs. So these plasmids will contain the genes for these transcription factors. Uh,
So can we
is
call
there
them
a
cloning vectors? Like all the fours, like all four can be used as cloning vector.
that is an not cloning vector, it should be an expression vector. Cloning vector just increases the number of the DNA
Oh,
copies.
right. Like can be used as vectors.
Yes, they can be. In this case, they can be because you want the transcription factors. So then there could be
Hmm.
mini circles, episomal, like you have some episomal DNA. I mean, that doesn't integrate again. Uh, they produce these, sorry, they produce these transcription factors, and there are some transposable elements also which go and they produce. So the idea is same, that you need to get the transcription factors in your cell so that you can produce iPSCs. Again, there are pros and cons for this, which I would suggest you all to study. And then there are non-viral DNA-free approaches also. Like we can use, small molecules, microRNAs, modified mRNA, and recombinant protein. To understand this figure will give you a good idea. So like microRNAs, as you can see here, they can be, inserted into the cell using liposomes, and then they could essentially they can form complexes which will inhibit certain genes. That could be done. Some episomal plasmids can be inserted through this route. They can, cause transcription and translation into, pluripotent proteins. Then there are, viral vectors like Sendai virus vectors, which are again non integrating vectors. There could be recombinant proteins, which could be used. You are directly inserting the protein that you want, that you can do. You can insert the mRNA. So again, mRNA vaccine, anybody knows SARS-CoV-2 ka mRNA vaccine? We already have Showman here. I think he will be able to answer these questions very well. He's working in Zydus. We'll come to you, Showman, just in 2 minutes. We have just a couple of slides. The idea behind this is we can transfer these genes in the form of DNA, RNA, or protein into the cell so that they become pluripotent. That's the idea. So again, different gene delivery approaches have been compared, and their efficiency and safety have also been compared. I would suggest you to read these papers if you want, on your curiosity, to understand which of these approaches are better, which one is not. Uh, so idea is, again, somatic cells will be taken, they will be reprogrammed to form iPSCs, and then they form they differentiate to form your cell of interest. So they can be used for regenerative therapy, tissue engineering, cell-based therapy, drug screening, disease modeling, and developmental biology. So this is the entire figure. It says the same thing. What are the different ways you can use stem cell? And then we have the different applications, as we already said. So Professor Shinya Yamanaka is still a very revered scientist. Still, he's alive actually. So who has created this stem cell technology, and it has a great potential. So we already have Showman here. I would request him to please interact with the students. Just again, giving you his introduction. He is an assistant manager at Zydus Life Sciences, Batti in Himachal Pradesh. So Showman, please, are you here? Can you hear me?
Hello, Shankita. Good morning, everyone. How are you,
Can you?
everyone? Am I audible, Shankita?
Good morning, sir.
Yes,
Am
you're
I
audible.
audible? Yeah,
Yes,
yeah.
yes, yes.
So right now, I'm in production department in Abadi site. So if you have any questions related to your academic, I mean, I'm taking pharmaceutical science and engineering, or production related, I mean, manufacturing related, please feel free to ask me.
I think there was one question previously. How is pharmaceutical engineering different from biotechnology? Maybe you could start with that.
Okay. See, this MTech Pharmaceutical Science and Engineering, this course is newly launched by 2020, and this course provides the training and development necessary to meet demand for highly skilled scientists, or you can say technocrats, within the expanding and national and global pharmaceutical sector, or any other sector like marketing or IT. Just like say, for example, my one of my friend, he she did BTech in biotechnology from HITK. Then uh, she completed MTech in pharmaceutical science and engineering, and she got a job Accenture Japan. So this course actually
Who is she? May
expand.
I ask?
Yeah, BTech in biotechnology from
Who is
HITK.
she?
Piyasha Dolui.
Oh, Piyasha. Okay.
Okay, and this course also lead to a wide range of careers, including academic, industrial, and healthcare applications of pharmaceutical science. Suppose if you want to pursue PhD in pharmaceutics or pharmacokinetics, pharmacology, so there is a huge scope. And or if you want to go with the industrial, just like say for example for me, and right now I'm in production department, so you can pursue this course. This course is very good, and this course is interdisciplinary course and designed to respond to increase the demand for estimate trained professional chemist. If you will see to next year as a chemist, so you took this course. This course is good, and research also they are and into commercially ready medical essentials and drugs that can be used for human well beings. So this is a basic overview of the MTech in pharmaceutical science and engineering, right? Or MTech in biotechnology, that I don't know actually, Sanket, the better to tell what are the course curriculum in there in MTech in biotechnology. So Sanket, the please share.
No, I think they know much about
Okay,
biotech
okay,
as of now.
okay.
But anyway, anything else? Anybody else would like to ask? Maybe Pragya.
Yes, yes, please, please.
This is the time that you could ask. You could
Acha, these
meet an
people are
industry leader.
BTech third year people or BTech fourth year?
BTech third year actually.
Third
BTech
Okay,
year.
fourth
third
year
year.
is BTech fourth year is horrible in heritage right now. So
You,
I
why?
think
So
they do not.
what are the career opportunities?
Career opportunity है ना? Suppose if you want to pursue in a PhD in a pharmaceutical sector, so then this course is very good. There is a huge scope for research, or you can say if you want to pursue the chemist, professional chemist, right? Or if you want to a job, just like say for me, ठीक है, तो ये भी course अच्छा ही है ना?
And also, sir, like, why would people like or the hire those who will hire, they will prefer a MTech in pharma than an MSc in pharma, like M pharma or whatever it is called.
Yes, yes. First of all, this course, I already told you, this course is newly launched by 2020. 2020. So this course, of course, it will difference from M.Pharm in pharmaceutics and M.Tech in pharmaceutical science and engineering. Suppose if you see those who are passed out from M.Pharm, so their salary actually very less compared to M.Tech in pharmaceutical science and engineering. You completed your course from IIT, then your package, I mean, should be more than 8 lakhs. ठीक है, तो ये एक्सपेक्ट तुम कर ही सकते हो. तो पैकेज का एक डिफरेंस आ सकते हैं, और क्या है जो कोर्स करिकुलम है, जो सेटअप है, ये डिफरेंट है बहुत. There is a huge difference from M.Pharm and M.Tech pharmaceutical science and engineering. Say, for example, if you want to, if you go to pharmaceutical science and engineering, there are the syllabus like, say, for example, computer-aided drug designing, unit operational, numerical analysis methods in chemistry, pharmacology and drug designing, formulation and drug delivery systems. So these are the subjects are not in the M.Pharm. There are the subjects like M.Pharm in pharmaceutics, pharmaconomics, pharmacological, regulatory and drug affairs. So ये actually difference है दोनों में, M.Pharm and M.Tech, because this M.Tech in pharmaceutical science and engineering course is an interdisciplinary course.
Okay, Shobin, one more thing. I have a question. I was having a question just a while ago. Since you are in production team, anything from maybe not revealing anything confidential, probably, but from anything from the recent advancements like gene therapy or things like that, maybe iPSCs or personalized medicine. If do people work in that kind of field in your company
So
or maybe some allied company?
this is basically R&D, research and development part. Production part,
Okay.
basically you have to manufacturing the tablet capsule, how there is the
Okay. Mm.
process and cycle time, and how they can make the tablet and capsule. So this is a basically research and development part. So this I don't know actually what else going on the research R&D part. So this is the difference between R&D and production.
Okay. 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 Uh, is
And so why?
okay. Shinjan, yes, please.
No, sir, you complete then I ask.
No, my another question was about the pharma companies in the US compared with the pharma companies in India. What is the difference?
Difference is the manufacturing facility, right? And the technological part. Suppose, say for example, if you see the in India, just like say for example, my company, Gadget Life Sciences Limited. So if you want to go to the soft floor, na, so if you see lot of process or lot of scope and equipment that are not technological advanced, and if you just go to the Pfizer or AstraZeneca, so there the process, there is the manufacturing facility. So there's a huge difference, and there to calculation or overall equipment efficiency of the equipment and cycle time of the product and their manpower. So this is the different lot of things that are actually present that I not discussed.
Okay. Anything else? We are seeing a very feeble response today.
So why did you choose this like after BTech?
Actually, my I cracked the gate examination. My gate rank 1140. So that time. So मेरे को एक शौक था कि आईआईटी से एमटेक करना है तो ये जो रैंक था मेरा 1140 तो उसी हिसाब से मेरे को एमटेक इन फार्मास्यूटिकल्स एंड इंजीनियरिंग फ्रॉम आईआईटी आइशन धानबाद से मिल रहे तो मैंने इसलिए ले लिए थे ये कोर्स
Was
मेरे को भी
NKJ sir
जाना
an
था एमटेक इन
inspiration behind
बायोटेक्नोलॉजी भी
this?
एमटेक इन बायोटेक्नोलॉजी भी जाना था बट मेरे को आईआईटी मिल गया था मैंने ले लिए थे उस टाइम आईआईटी का टैग है ना तो उसका
लाइक उस
एप्लीकेशन भी
समय
हो
आपका
गया जॉब
क्लियर
मिल
था
गया
कि
और
इसके बाद क्या
कैसे
क्या कर सकता?
आफ्टर completing my this job.
Was it clear to you
Hello.
right from the beginning that you need to do BTech, then MTech, and then you want to go to industry? Was it
Yes,
clear right from the BTech?
yes, of course. Why not? मेरा जो टारगेट है ये है अभी तो असिस्टेंट मैनेजर हूँ आफ्टर 5 to 6 years to see myself as a senior leadership team member जस्ट सीनियर मैनेजर डीजीएम जो कंपनी का जो हायर पोजीशन होता है सीनियर लीडरशिप टीम यू नो सीनियर लीडरशिप टीम सिंजन
यस सर.
you know what are the designation in a senior leadership team
Not
designation
clearly, sir.
डेजिग्नेशन पता नहीं क्या क्या है
Not sir.
I
सबसे
also
लोएस्ट
don't know
होता है
actually.
ओके ओके
Yeah, Showman, please go ahead.
फास्ट है अपेरेंटिस जो डेजिग्नेशन है जब से स्टार्ट होता है ये एक्चुअली कॉन्ट्रेक्चुअल बेसिस में होता है अपेरेंटिस जो लोग लेते हैं ये कंपनी फार्मास्यूटिकल ये फार्मा कंपनी तो कॉन्ट्रेक्चुअली उसका सैलरी बहुत कम होता है जो मिनिमम है 12,000 से स्टार्ट होता है अपेरेंटिस के बाद होता है ट्रेनिंग ट्रेनिंग के बाद ट्रेनिंग ऑफिसर देन एग्जीक्यूटिव देन सीनियर एग्जीक्यूटिव देन असिस्टेंट मैनेजर देन डेप्यूटी मैनेजर देन एसोसिएट मैनेजर then DGM, then CTO, चीफ टेक्निकल ऑफिसर। तो मेरा विजन यही है, आफ्टर 5 to 6 years, माई सेल्फ DGM और CTO।
So,
क्लियर है
what
समझो?
is
salary?
सैलरी जो CTO का जो सैलरी होता है वो
Yes, sir.
अभी तो जाइडस में है 3.4 करोड़।
अच्छा।
एकदम
okay.
सीधा एकदम सैलरी में आ गया सृंजन। देखो सृंजन, फर्स्ट
Yes,
तुम्हारा
sir.
टारगेट क्या है? अभी 3rd ईयर चल रहा है ना, तो नेक्स्ट ईयर क्या करोगी? अच्छे से गेट क्लियर करो, ठीक है? गेट होने के बाद कोई IIT NIT मिल गया तो ले लो, ठीक है? IIT NIT का टैग टैग क्या संकेत था? एक सिग्निफिकेंस तो है IIT का टैग।
Sorry,
IIT NIT का टैग का तो एक सिग्निफिकेंस तो है इसे।
I don't. It's no. It's not there in IITs at least, but it's there outside IIT. That I know. So yeah,
ठीक
yeah.
है,
So
कोई नहीं।
yeah. Okay, I think that would be. Is there any more questions? I don't think so. Nobody is responding
देखो क्या है,
anymore.
सपोज इफ यू हैव एनी डाउट और to set up your future, please feel free to contact my LinkedIn. Okay, I will definitely reply you.
Sir, LinkedIn नाम?
LinkedIn नाम Showman with
You could
SOUMEN।
also ask NKJ sir. He was NKJ sir student, so he will proudly give his contact.
यही तो प्रॉब्लम है सर।
Okay. Okay, Showman, thank you very much for joining us. We will, I mean, you can leave if you want
So
to.
there's one more question, sir, which
Okay.
my friend was asking
Okay,
to
okay.
ask you. So that ওই দিদিটা কি করছে যে অ্যাসেঞ্চার জাপানে জিজ্ঞেস করছিল, লাইক বলছিল।
ও তো এখন ওর অ্যাকচুয়ালি প্রজেক্ট আছে, তো ও এমটেক করেছে আমার সাথে। she completed her MTech in pharmaceutical science and engineering. She will be my batchmate, and she will join probably November. So their
Okay, sir.
profile will be the project, I mean the business analyst.
Achha, sir, I had one question. Like after this MTech in pharmaceutical, if an MBA degree does it like boost the CV or something like that? Like, is it
MBA
required for
degree,
it? Yes,
MBA
sir.
degree.
In
Yes, yes,
industry.
yes, of course. Yes.
So, sir, মানে যারা MTech এর পর MBA করছে তারা actually বোকা না।
No, no, no.
একদমই না, একদমই না, একদমই না।
এমটেক এর পর যারা পিএইচডি করছে তারা বোকা। এমবিএ করছে তারা তো বোকা নয়।
যারা PMR তারা নয়।
I can't say that. Anyway, really, Showman is one of the I would say, as I already said, industry leaders, and in India at least he's in the top 5% of the population, maybe top 5% or maybe even less. It would be less, not more.
अरे नहीं नहीं
So,
नहीं, ऐसा नहीं, कुछ भी बोल सकते हो तुम।
so it's like that. So that's good. You're still in your office?
यस यस, सैटरडे
Great.
भी छुट्टी नहीं है संकेत दा।
I mean, I thought the
तो
students will be
सचिन
very
को
much,
पढ़ेगा कब क्या?
very much
ये प्रोडक्शन एरिया
enthusiastic
है
to
भाई,
eat.
देख लो, मैं
I
प्रोडक्शन
think
एरिया
you're
में हूँ अभी।
demotivating them. I don't know. it's it's anyway, but I can see he has changed. Although he had this leadership thing always. So he used to organize all those, heritage utsav and all that. He used to be there in the front line of, people who used to do protest in front of Arvind sir. But now things have changed.
अभी सिचुएशन क्या है? फ्रेशर्स वगैरह हो रहा है कुछ? फ्रेशर्स বাধ্য হয়ে গেছে
ইয়েস স্যার, তো ফেস্ট আমাদের মানে ফ্রেশার্স আমাদের হয় এক্স্যাক্টলি একদিন আগে ক্যান্সেল হয়ে যায়, বাকি
ওটাই তো
সময়
হয় প্রথম থেকেই, তো এটা তো হেরিটেজে এটাই নিয়ম, এটাই রুল।
এটার
আপনার
ব্যাপারেও
স্যার
কিন্তু সৌমেন খুব ভালো আর কি সাজেশন দিতে পারবে
করানোর জন্য না না করানোর জন্য?
করানোর জন্য, মানে যদিও
আচ্ছা,
এনি উই উইল ডিসকাস সেপারেটলি, টেস্ট রিসার্চ এবার।
ওকে ওকে ওকে, এনিথিং এলস?
অর এনিথিং এলস?
By the way, IIT Delhi is having its fest from 5th to 8th October. Anybody wants to come, they can come in. Showman, you can also come in.
দিল্লি
Rendezvous
অনেক
X.
দূর।
ছুটি নেই গো।
স্যার স্যার
Okay.
অফিস ট্যুর প্লিজ একবার।
কি office tour? Okay,
হ্যাঁ স্যার
okay.
একবার দেখান
He's
না,
telling me.
শোভন স্যার একবার
কোথায়?
অফিস ট্যুর
Office
স্যার
tour
আপনার।
দিতে বলছে।
কি
এখন
হয়? হ্যাঁ স্যার, মানে কি হয় জানতে চাই।
দেখ, প্রথম কথা হচ্ছে এখানে আমি তো প্রোডাকশনে আছি। এখন হয়তো তোদের মাথার মধ্যে জিনিসগুলো ঢুকবে না, সেটা হচ্ছে যে এখানে যেটা হয় ট্যাবলেট ক্যাপসুল প্রোডাকশন হয় এখানে। তো আমি তোদেরকে এই জন্য ডিটেলসে বলিনি, কারণ যখন তোরা এই কোর্সটা নিবি তখন জিনিসটা বুঝতে পারবি যে অ্যাকচুয়ালি কি হয় প্রোডাকশনে। প্রোডাকশনে কি হয়, তো ম্যানুফ্যাকচারিং এ তো বিভিন্ন ডিপার্টমেন্ট আছে, দেয়ার আর দ্য ভেরিয়াস steps like granulation, compression, coating, inspection, right? You heard this term,
Yes sir,
granulation,
yes
compression,
sir.
coating. तो क्या है, तो ग्रेनुलेशन में देर आर लॉट ऑफ machine, जस्ट आरएमजी है, रैपिड मिक्स्ड ग्रेनुलेटर, जहाँ पे क्या है एपीआई और जो एक्सीपिएंट है ना वहाँ पे मिक्स्ड होता है आरएमजी में फर्स्ट। देन क्या है, बाइंडर एडिशन होता है, बाइंडर में सपोज मैग्नीशियम स्टीराइड है, सपोज प्रोडक्ट टू प्रोडक्ट वेरी होते हैं ये। देन ये ड्राइंग के लिए एक एफबीडी है, फ्लूइड बेड ड्राइंग और फ्लूइड बेड इक्विपमेंट, वहाँ पे ड्राइंग के लिए जाते हैं। तो हर एक प्रोडक्ट के लिए ड्राइंग टाइम अलग अलग है। देन ड्राइंग के बाद ये ब्लेंडिंग होता है, सारे जो एपीआई एक्सपीएंट और जो ब्लेंडिंग जो बाइंडर सॉल्यूशन आती है ना, वो सारे क्या ब्लेंडिंग में वो बाइंड होता है। देन ब्लेंडिंग के बाद वो कॉम्प्रेशन में जाते हैं। तो बीच में बहुत सारी इन प्रोसेस चेक भी है, जो एवरेज वेट है, फ्रेबिलिटी चेक, हार्डनेस, वो कॉम्प्रेशन के बाद ही होता है। देन आफ्टर कॉम्प्रेशन, देन सपोज किसी किसी प्रोडक्ट में फिल्म कोटिंग होता है, शुगर कोटिंग, ये डिफरेंट है, प्रोडक्ट टू प्रोडक्ट वेरी होता है। तो कोटिंग के बाद तो इंस्पेक्शन में जाता है टैबलेट वगैरह। तो इंस्पेक्शन के बाद पैकेजिंग। तो देयर आर लॉट ऑफ प्रोसीड्यूर, लेंथी प्रोसीड्यूर। बीच में क्लीनिंग है, टाइप ए क्लीनिंग है, टाइप बी क्लीनिंग है। ये टाइप ए क्लीनिंग सपोज बैच सेम बैच होगा तो टाइप ए क्लीनिंग होगा, बैच डिफरेंट आ जाएगा तो टाइप बी क्लीनिंग होगा। तो सारे जो इक्विपमेंट जो पार्ट्स है उसको डिसेंबली होगा, देन ज्वाइन होगा, बहुत सारे लेंथी प्रोसेस है। और जो इन प्रोसेस चेक मैंने बताया ना, जो फेरेबिलिटी चेक, हार्डनेस, ये वेट वेरिएशन, डिसोल्यूशन, डिसइंटीग्रेशन, इसमें भी बहुत टाइम होता है। तो देयर आर लॉट ऑफ प्रोसीड्यूर्स होते हैं। तो अंदर
चलिए
रिस्ट्रिक्शन भी है, कैमरा वगैरह कुछ अलाउ भी नहीं है अंदर।
Okay. It's
बोलो।
difficult to record that. That
यस
hence.
यस यस।
Okay.
like each step
तो
has
मैं
a
प्रोडक्ट,
checkpoint kind of thing।
करेक्ट, चेक पॉइंट।
তো ওই চেক পয়েন্টে দেখা যায় না?
চেক পয়েন্টে হ্যাঁ একটার পর একটা জাস্ট কোয়ালিফাই হবে, টিক ঠিক হবে, তারপরে নেক্সট প্রসেসে যাবে। সাপোজ গ্র্যানুলেশনে সাপোজ অল ইজ ওকে টু বি চেক বাই আইপিকিউসি, जो क्वालिटी कंट्रोल আছে ডিপার্টমেন্ট আর
ওকে।
কোয়ালিটি এসপিডেন্স, তারা গিয়ে চেক করবে তো সমস্ত কিছু ঠিকঠাক আছে কিনা। দেন তোর সমস্ত ট্যাবলেট বল সেগুলো নেক্সট স্টেপে যাবে। আবার কম্প্রেশন হওয়ার পরে আবার ট্যাবলেট যাবে আইপিকিউ এর পাশে, ওরা চেক করবে যে হ্যাঁ সবকিছু ঠিক আছে কিনা, প্রোডাক্টের মধ্যে কোনো ভ্যারিয়েশন নেই কিনা। দেন তারপরে ওটা কোটিং এ যাবে। তো এটা একটা অনেকটা চেক লিস্টের মতনই, যে দেখবে যে হ্যাঁ এভারেজ ওয়েট যেটা আছে সেটা তোর লিমিট আছে বিএমআর এর মধ্যে, সেটা ফিক্সড আছে। যে ওয়েট ভেরিয়েশন, ট্যাবলেটের থিকনেস, যে এই সাপোজ প্যারাসিটামলের যে ট্যাবলেটের যে ওয়েট আর ট্যাবলেটের যে ভেরিয়েশন, যে ফ্যাবেবিলিটি, ডিজলিউশন টাইম, এগুলো সব মেনশন করা আছে ওখানে বিএমআর এর মধ্যে। ঠিক আছে।
তা চেক
ওরা
কিভাবে
জাস্ট চেক
করো?
করবে যে ওই লিমিটের মধ্যে। হ্যাঁ,
ওইগুলো চেক
সাপোজ
কিভাবে
ধর
করো?
হার্ডনেসের জন্য ট্যাবলেটের হার্ডনেস চেক করার জন্য মানে আলাদা করে মেশিন আছে। ফ্র্যাবিলিটি চেকের জন্য আলাদা মেশিন আছে, সঙ্গে আলাদা আলাদা ইকুইপমেন্ট আছে, ডিজলিউশন, ডিসইন্টিগ্রেশন। তুই সার্চ কর গুগলে যে ডিজলিউশন ইকুইপমেন্ট, ডিসইন্টিগ্রেশন ইকুইপমেন্ট, ফ্র্যাবিলিটি, হার্ডনেস, ওগুলো চলে আসবে অটোমেটিক্যালি। ওখানে ওগুলো চেক হয় সবকিছু। হার্ডনেস টেস্টার আছে, যেখানে তোর ভার্নিয়ার ক্যালিপার দেখেছিস না, ওখানে কি হয় থিকনেস মেজার করা
হুম।
হয় ট্যাবলেটে। ঠিক আছে। যে থিকনেসটা ঠিক আছে কিনা নেই। যদি সাপোজ কোনো ডেভিয়েশন বা কোনো কিছু চলে আসে তখন সেটাকে আবার রিচেক করা হয়। বা অনেক সময় কি হয়, পুরো ব্যাচটাকেই পুরো ফেলে দেওয়া হয়, কারণ এটা একটা ইউএস এফডিএ অ্যাপ্রুভাল প্ল্যান, রাইট?
মানে একটা ভুল হলে পুরোটা ক্যান্সেল করে দেওয়া হয়।
হ্যাঁ হ্যাঁ হ্যাঁ, সাপোজ আমি তোকে একটা এক্সাম্পল দিচ্ছি। কিছুদিন আগে, লাস্ট 2 সপ্তাহ আগে পুরো প্রসেস হয়ে গেছে। প্যাকিং এর যে প্যাকিং হয় না, প্যাকিং এর বাইরে যেটা তোর এক্সপায়ারি ডেট লেখা থাকে, সেটা 2025-এর জায়গায় 2024 প্রিন্ট হয়েছিল। ঠিক আছে, তো তোর 12,40,000, 12,40,000 এর তো ট্যাবলেট ছিল, পুরো ডিস্ট্রয় হয়ে গেল পুরো। মানে ওটা একদম ইয়ে থেকে মানে ব্যাড ট্যাবলেট হিসেবে কনসিডার করা হলো, প্যাকিং কে বাদ। মানে ভাবতে পারছিস,
জাস্ট
তো
প্রিন্টিং
যেমন
এর জন্য।
প্রফিট আছে কোম্পানিতে সেরকম। হ্যাঁ বল।
আর আর একটা জাস্ট এক্সপায়ারি ডেটটা কিভাবে তৈরি করে, লাইক বলে, লাইক আফটার
ওটা
প্রোডাকশন।
ফর্মুলেশন আর&ডি টিম ডিসাইডেড করে, ওটা আর&ডি টিম ডিসাইড করে যে
আচ্ছা।
That's
একটা
why
এপি এর
researchers are
মধ্যে কোন কোন।
important, Shinjan.
ইয়েস, অফকোর্স, রিসার্চার্স ইজ অলসো ইম্পর্টেন্ট। অ্যান্ড জাইডাসের আর&ডি টিম আছে আলাদা আহমেদাবাদে, যারা এই সমস্ত ডিসাইডেড করে যে কোন এক্সিপিয়েন্ট কোন এপিআই এর সাথে মিক্সড হবে, ঠিক আছে, কতটা অ্যামাউন্টই মিক্সড হবে, কতক্ষণ ড্রাইং হবে। যে আমি তোকে বললাম না, ড্রাইং হয় এবিডি এবি এর
হুম।
মধ্যে, তো ড্রাইং টাইম ওখানে স্ট্যান্ডার্ড ফিক্সড আছে। যে সাপোজ প্যারাসিটামল, তো ড্রাইং টাইম 60 মিনিটস। যদি 60 মিনিটস এর উপরে তোর যদি এলওডি, লস অফ ড্রাইং যদি না আসে, তাহলে সেটাকে তো ডেভিয়েশন ব্যাচ হিসেবে কনসিডার করা হয়। ঠিক আছে, বহুত সারি প্রসিডিউর আছে, আস্তে আস্তে জানতে পারবি। ঠিক আছে, যদি আসিস আমার সাথে যোগাযোগ করিস, কন্ট্যাক্ট, সবকিছু বলে দেবো।
তা আপনাদের ওখানে ইন্টার্নশিপ হয়? আমার একটা তো
ইন্টার্নশিপ হয়, আমি তো ইন্টার্নশিপের থ্রুতেই পেয়েছি এখানে। আমি তো লাস্ট ইয়ার 2022 Zydus came our college for internship purposes. So they took our interview. Then I am the only person who was selected. Then internship के बाद তোর একটা ফিডব্যাক ফর্ম এসেছিল আমার কাছে যে হোয়াট অফ ইউর এরিয়া অফ ইন্টারেস্ট, তো ম্যানে সিলেক্ট কিয়া প্রোডাকশন। তো দেন প্রোডাকশনের যে হেড আছে রামরাজ, যে মানে আমার যে প্ল্যান্টের হেড, উনি ইন্টারভিউ নিল তোর। দেন তোর পেয়ে গেলাম। আমি তো ইন্টার্নশিপ করেছি 6 মাস। 6 মাস ইন্টার্নশিপ করেছিলাম তোর হচ্ছে এইচআর ডিপার্টমেন্টে, জাইডাস স্কিল ডেভেলপমেন্ট সেন্টার, যেটা আহমেদাবাদে আছে।
আচ্ছা।
ঠিক
I
আছে,
think all the technical questions are done. Just few questions more, Shoman, just one or two. How is the food at Himachal Pradesh?
নট গুড।
And have you been going to Kullu, Manali, Shimla, those places?
No, 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 no. Right now
Not
the
yet.
situation is yes,
Not
situation
good.
so there are a lot of bridges are destroyed.
And another, just one question. I think that would be the end. So, are you coming to Kolkata during Puja?
No, no.
No.
I will come Kali
আচ্ছা আচ্ছা, ঠিক
দুর্গা
আছে।
পূজা में ছুটি নেই।
ছুটি দেয়নি কি ছুটি পাসনি? মানে দিবে
ছুটি
তো
নেই
না,
গো।
মানে কর্পোরেটর ছুটি
না না,
দেয়।
ছুটি মুটি নেই কিছু। আবার এই প্রথম বছর না, ফার্স্ট বছরে কোনো পিএল নেই, সব এসএল আর সিএল আছে। তো এসএল আছে 5টা, সিএল আছে 5টা, সেটাকে আমি একসাথে আবার অ্যাপ্লাই করতেও পারবো না।
আচ্ছা, মানে ওটারও পলিসি আছে আলাদা।
হ্যাঁ হ্যাঁ, শুধু পলিসি পলিসি পলিসি, টাকা যেমন দিচ্ছে সেরকম পলিসি ভরে রেখেছে চারিদিকে।
ওকে।
না ভালো ভাই, তোরা পড়াশুনো কর, ঠিক আছে, গেট টেট কোয়ালিফাই কর, আইআইটি থেকে পড়। সাপোজ তোদেরকে মার্কেটিং এ যদি যেতে চাস, প্যাকেজ খুব ভালো। যদি কোডিং টোডিং পারিস করতে, ঠিক আছে, এই যে পিয়াসা যে নাম বললাম, ও ভালো কোডিং অর প্রজেক্ট ম্যানেজমেন্ট অর পাইথন মেশিন লার্নিং ও মোটামুটি বেসিক জানেন জিনিসগুলো। ঠিক আছে, তো ওই জন্য পেয়েছে ও। তো তোরাও চেষ্টা কর, পেয়ে যাবি। ঠিক আছে, যদি হায়ার প্যাকেজ যেতে হয়, দেখ আইটি একমাত্র তোদেরকে বেশি পয়সা দেবে। ঠিক আছে, কি সংক্ষেপে এগ্রি করো না ইন ইন্ডিয়া।
না না, অফকোর্স অফকোর্স, আমাকে নিজেকে কোডিং
ঠিক
করতে
আছে,
হচ্ছে, মানে আমি অ্যাজ এ রিসার্চার কোডিং করছি, মানে দ্যাট ইজ দ্য ওয়ার্স্ট কেস সিনারিও আই থিঙ্ক।
ঠিক আছে।
যে তো
তো তোরা যদি
মানে এটা তো কিছু করার নেই, মানে কোডিং হ্যাজ টেকেন আপ আর এন্টায়ার দিস lot of jobs also in that aspect. So thank you, Showman, for joining. I think that would be all from your end. You may leave if you want to, you may stay if you want to. That's all. Just a few announcements that I have for the students. So today was the last lecture. Thank you all for joining the course. So since you know, we have the presentation assignment on 16th of September from 3 p.m. So please fill up the the sheets that has been given. The final quiz will be on 30th September at 11 a.m. That is 3 weeks from now. It will be totally MCQ based. So you have 3 weeks to prepare. So I hope that you have spent a nice time learning about aging. I hope this course was informative and enjoyable. And I sincerely wish that each of you progress very well in life. You find success wherever you want to, find success in, whether it is in research, industry, education, or any other endeavors. Remember that aging is a natural part of life, and it is never too early or late to start taking care of yourself and planning for future. So keep learning, growing, striving towards your goals. With that thought in mind, this is all that I have to offer to you on this course. I hope that I meet you all soon on campus. Any doubts? Anything else? I think there were a couple of questions. Antik has had a few questions from slide 40 and 41. Are these the slides?
Okay, Shankit, bye.
Yeah, you may leave.
Wish you all the best.
Soumit,
Yeah, thank you. Thank you, everyone.
what was your question?